Morning, everyone. We've waited the requisite 2 minutes that I think is modern current Zoom etiquette, I guess, to allow people to come in. So welcome to our BME open house event around Capstone, and particularly Capstone projects dealing with disparities. We have 4 really outstanding Capstone teams that are going to be talking about what they're working on. Just to give a little background, though, about Capstone. So all biomedical engineering majors have to complete their curriculum in their 4th year with a Capstone project. And this is a year-long project. We're very broad in the kinds of projects that are allowed, and as long as they are new, it's not something that just has been done before, as long as it somehow pertains, very broadly speaking, to health in the biomedical realm, and as long as it's year-long in scope and has some sort of design aspect, that constitutes a Capstone project. And you can see you'll see the range even in the 4 teams that we have here is quite broad in the kinds of work that's being done and the nature of these projects. Projects are not just mentored by biomedical engineering faculty, although many of them are. Just this year alone, I was doing some tally right before this meeting. We do have quite a number of projects that are advised by biomedical engineering faculty. But as far as our advising pool, both advisors and co-advisors or co-mentors on these projects, we have represented 4 non-BME departments, 5 basic science departments, most of those in the School of Medicine, but I think also we have chemistry. We have at least 2 or 3 that are not even UVA, they're other academic institutions, including one of the speakers today. 13 clinical departments, so otolaryngology and cardiovascular medicine, orthopedics, but just this year alone, we have 13 clinical co-mentors and 10 companies outside of BME. So it's really a wide variety of the kinds of projects that are done. Students who have worked in Capstone have gone on to present their work at national meetings, become co-authors on publications, get provisional patents submitted, and then sometime within like a couple of years down the road, I know of one that just issued recently for a student who graduated in 2015. So almost 6 years down the road, got a full patent issued for a startup company that he's a co-founder on. So again, they've gone on to do really wonderful things with this work in terms of what has been accomplished for capstones. This year, Capstone is a little bit different in that students predominantly have not been coming in person, with some exceptions, to work in research labs, as has happened often in previous years just because of constraints due to COVID in terms of School of Medicine research opportunities. But all of the Capstone teams and the advisors have really pivoted wonderfully, I think, to a virtual process. Hopefully this will be the last year that this will be necessary, but it's still, I think, I'm personally really wowed by the creativity of all of our students and the resilience of all of our students to be able to do this. So with that introduction, is there anything that you wanted me to add or you would like to add, Shannon, in terms of, so we have 2 Capstone instructors. I teach the fall semester of Capstone, and Professor Barker, Shannon Barker, is the instructor for the spring semester of Capstone. So both of us know all of these students very well.
Yeah, I think you covered it really well. I would just echo the pivots, these really innovative pivots that were made to do this in a pandemic year. So the fact that the students were able to accomplish what they accomplished, given these limitations, just really goes to show how talented and innovative they are. I also want to add one last thing, is that at the end of the 4th year, all the Capstone teams get to present their work to the broader BME community. And it's really a time of celebration. So the BME community comes together and celebrates everything that these students accomplished. It's a really important milestone for a BME student, and that's always really fun. So should I go ahead and start?
Yes, you're
right.
Introducing our first 2 speakers.
Yeah, so I have the honor of introducing the first 2 teams. The first one we'll hear from today is a team that includes Simone Heron, Hunter Newland, who's going to be presenting, and Kunal Sarnik. And they worked with Dr. Sean Moore in UVA Pediatrics here. The title of their project is Structural Racism as a Barrier to Inactivated Influenza Vaccination Coverage During Pregnancy and Influenza Spread Modeling in the Americas. So this is a really wonderful example of biomedical engineers using engineering tools to address a glaring healthcare disparity, in this case using data science. And the goal of the project was to develop a tool that may be used to create better influenza vaccination campaigns for underserved populations in both Brazil and Virginia. So really excited to hear from Hunter. Hunter, take it away.
Sure thing. Thanks. Oh, can you enable screen sharing, please? Thanks. Can everybody see that okay? All right, awesome. So yeah, as Dr. Barker said, this is our project. My teammates couldn't be here today, but that's perfectly fine. The title of our project is Structural Racism as a Barrier to Influenza Vaccination Uptake During Pregnancy in the Americas. We focus on Brazil, the country of Brazil, and a little bit on the state of Virginia. So our project deals with influenza during pregnancy. So during pregnancy, there is a higher chance of contracting the flu, which can lead to low birth weight, low APGAR scores for newborns, higher chance of infant respiratory infection, worse complications down the line in relation to infant respiratory health. There's a higher rate of maternal morbidity and mortality associated with it. And so we have been trying to quantify and analyze the rates at which these outcomes negatively impact certain groups over others, particularly Black and Brown groups in Brazil versus the white population, pregnant population. And similarly for Virginia, we're trying to elucidate the reasonings behind influenza uptake or not for the Black pregnant population in comparison. So to the white pregnant population. And some of these inequalities have already been measured. For instance, you can see right here in this figure from the CDC, the darker the blue, the less likely that there will be an uptake, like the lower the percentage of influenza vaccination uptake. So for instance, in Virginia, the lowest rate for all of the racial populations in the pregnant category is non-Hispanic Black pregnant women. And they are 30% less likely to receive a flu shot due to multiple factors, including misinformation about influenza vaccines, the history of doctor-patient mistrust, and the lack of like insurance coverage or related to their socioeconomic status. And we're trying to statistically prove that for the country of Brazil and elucidate it in Virginia. So our project is broken down into two separate pieces. So we have a quantitative and a qualitative project. Our quantitative focuses on doing a influenza spread model for Brazil. We're using computational models, so a lot of modeling packages in R, like EpiModel. We're trying to model it sort of after the Biocomplexity Institute's model for Zika. So that's really interesting. You should check that out if you're interested. And so we're doing it based on race and pregnancy status for the entire country of Brazil. And if you look over, this is kind of a tangent, but if you look over on the right here, this little blue country is Sierra, and we've done gestation period and birth weight data for all of the states, the little municipalities in Brazil, for the white, the Branca population, and the Pretos, which is the Black population. And you can see that there's like much more volatile data which is just like irregular gestational periods in comparison to these very like periodic rising and falls throughout the years for the white population. So we know that there is a disparity in between just pregnancy in general. And so we want to connect it to flu vaccinations and illness. And so that's basically what our model is going to do. Based off of that model, we will provide a scheduling vaccination recommendation based on seasonality and periodicity for each population group, because right now Brazil only has one vaccination campaign that spreads over a couple of weeks for the entire country. And that's just not how flu spreads. It starts in the north and goes south. So it negatively impacts a lot of particularly Black pregnant populations. And so that's our quantitative project. And then our qualitative project, we're doing some survey and interview questions in the Blue Ridge Health region of Virginia with 30 Black pregnant women or recently pregnant women about their experience with the flu or the flu shot. And we're just trying to gauge what the disparities are in relation to their like opinion of it, like their experience, their understanding of what's going on. So we'll use a lot of like health belief model tactics. We'll do some coding and some statistical analysis related to that. So hopefully we'll have a very well-rounded idea of the disparities, and then we can give some recommendations based on the data to improve these disparities for the country of Brazil as a whole, and then also in the Blue Ridge region of Virginia. So yeah, thank you for listening. And if you have any questions later, I'm happy to answer them. Thank you.
Wonderful. Thank you, Hunter. I said this in the text, but just remember that you can save your questions till the end of the 4 presentations. But if you want to go ahead and put your question in chat, that's fine too. And I'll ask the students once we get through all the presentations. So now I'm going to introduce the second capstone project. This one is led by Maria Sammaritano. She's working with 3 folks in the obstetrics and gynecology department at the Baylor University School of Medicine. The title of her project is Vagina-on-Chip Model for Elucidating Underlying Mechanisms of Vaginal Cell Interactions. This is a really good representation of how biomedical engineers can tackle issues in ways that reduce overall healthcare disparities and inequities. And that includes women. So the historical exclusion of women from the drug development process has led to a lack of information, right, about women's health, about women's physiology. And so the goal of her project was to design and establish the feasibility of a vagina-on-a-chip system to model 3-dimensional vaginal tissue physiology using microfluidics. So take it away, Maria.
Hello, everyone. I'm so excited to present this to you. My project is something that I'm really passionate about. Especially in the past year, I've learned a lot about women's health and disparities and gender bias in biomedical research, and it's kind of been my passion, what I want to go forward and pursue in the future. So I'm really excited to tell you about this project. So, as Dr. Barker explained, there's been a systemic overlooking and exclusion of women from biomedical research, really starting even at the cellular level with the use of male cell models and then male animal models. And so as a result, things are really just studied in male models for the most part, and then just extrapolated to women. And as a result, a lot of things that present differently in women or affect women exclusively or disproportionately are left undertreated or mistreated often, and it leads to poor outcomes for women in the healthcare system. So in particular, one of these gaps in research is research on vaginal tissue. There's really not many good models for studying vaginal tissue in a way that's clinically relevant. So as a result, a lot of vaginal conditions go without treatments, and a lot of women suffer in the healthcare system as a result. So my project is aiming to use concepts from the field of organ-on-chip technology, an exciting emerging field of microfluidics, to solve this problem and create a really clinically relevant 3D recapitulation of vaginal tissue in a device that's well controlled and precise and can really be used to study mechanisms of vaginal tissue, as well as drug testing and vaginal tissue down the line. So this is an image of my prototype. I've been prototyping actually a few different designs, but the main design criteria that I've been working on is the fact that the vagina is has multiple layers. So there's three main cell layers that make up the vagina, and thus it's kind of hard to study by just putting cells in a dish and studying the flat culture. It doesn't really give a good representation of how human tissue will actually respond in vivo. So I've designed this device that includes a cell culture chamber with enough depth to include hydrogels, with each vaginal cell type stacked on top of each other. And so the hydrogel will serve as a scaffold to help the cells grow into that 3D construct that really represents how it will be in a human. And so my design includes the cell culture chamber in the middle. At the end, there's a port for attaching a syringe pump to it so that you can pump cell media continuously and therefore sustain the cell culture for long periods of time. And then a reservoir at the end where you can collect the excess media and test it for different things like growth factors or different metabolic components and use that to further study vaginal tissue. So in this way, the Vagina-on-Chip can be used as a really scalable, effective, functional device, and that can then be used downstream for a lot of applications. Like you can include hormones and have an even better representation of vaginal tissue throughout the cycle because it changes in response to hormones throughout the menstrual cycle. Something that the lab that I work with virtually, the lab at Baylor does, they study vaginal wound healing. So we're actually writing grants right now to continue this project in person, and hopefully I'll be able to go there and continue working on it next year. But one of their primary things they want to study is mechanisms of vaginal wound healing. And right now it's not really helpful for them to just study the fibroblast layer in a dish. It's not really giving a good representation of what vaginal wound healing would look like in person. So this is what I've been working on, and yeah, I'd love to answer any questions later.
Thanks, Maria. Very cool.
All right. Sorry, I was having trouble finding my unmute button. So those are both great examples of projects. The first one that deal with inequities and by taking a data science and computational modeling approach, and what Maria just talked about, taking a cell tissue, almost bioreactor bioengineering approach to that. And the next project I'm happy to introduce are three students, Claire Reagan, Lauren Hewlett, and Aaron Whetstone. And Aaron, I believe, is going to be the speaker for this. And their project title is Sewage Surveillance Tool for Tracking of SARS-CoV-2 in Urban Bangladesh. This is obviously highly relevant right now still, particularly in Bangladesh. The medical infrastructure is just not really well equipped for its large population size in general, and the hospitals are really unprepared for the patient influx caused by SARS-CoV-2. And they really needed a more effective disease surveillance method there, which I know Aaron will talk about. And so the goal of their project is just to assess quantitative data for viral load from sewage, specific sewage lines, and to present this data in a dashboard that's available more broadly. And I just wanted to briefly comment on that. The dashboard that, by the way, their primary mentor is a faculty member in infectious diseases here at UVA. And this dashboard is actually monitored, and it's monitored daily, consulted daily by the president of Bangladesh. And to my knowledge, it's the first electronic dashboard in the world that's designed and deployed for a developing nation. So this is certainly very important in addressing a disparity in terms of developing nations relative to more wealthier nations that are a little bit better equipped and have more resources to address this infectious disease. So with that, I will turn it over to Erin.
Yeah, thank you so much, Dr. Allen. Let's see. Very excited to share this with you guys. But I guess just as a preface before I start, it has been very cool to kind of see my peers and my professors as well kind of encourage these projects that are targeting these health disparities and kind of see how we can use our academic and classroom knowledge for problems that really matter and need a little bit more attention. So anyways, yes, my team and I have been working to create the sewage surveillance tool for tracking SARS-CoV-2 in urban Bangladesh this year. So just a little bit of background behind Bangladesh. It's located right here to the east of India, and it hosts the capital city of Dhaka, which is one of the most densely populated cities in the world and has a population of approximately 22 million people. And it is also home to many urban slums, which are often tightly packed and also have high poverty rates in general. So you can imagine that a virus like SARS-CoV-2 can spread pretty rapidly in these communities and can also be pretty hard to track. And so that's why our advisors have rolled out this wastewater surveillance project, which is a really great solution to tracking viruses like SARS-CoV-2 because it is of no cost to the residents and has also been shown to be highly accurate. So this is kind of just a good infographic of what wastewater surveillance does and how it's conducted. But basically the wastewater samples are collected and then the viral load of the virus is quantified through a process called RT-qPCR. And then that viral load data can go on to inform public health officials of the prevalence and also help them make actionable health decisions about what locations need to be investigated further. And so to create a visual representation of that data, we have developed this COVID-19 dashboard for Dhaka that represents the 8 wards and 33 collection sites that our advisors work out of. And to kind of show how that works, I'll move to the online version. But yeah, so public health officials and other users of the dashboard can kind of move through the different dates and see how the virus has been spreading over time. We also have linear trends here. We have a current situation tab that kind of gives a snapshot situation of the most recent data for public health officials, just to have a quick summary. And then you can also track the linear trends of the viral load and case counts by ward and by sites. And so yeah, this has been our project this year, and we, yeah, it's been really great to be a part of, and also we are hoping to hand it over to their public health officials really soon. The Prime Minister of Bangladesh does use it for reference, but yeah, we are about one step away from getting it up on their website for everybody in the country to use. So yeah, and happy to answer any questions after everybody's presented as well. So thanks.
Wonderful. Thank you so much, Erin. Such a timely project. All right, and then our last speaker is Connor Emelung, who is working with a faculty member in the BME department on a project titled Creating Accessible Research Opportunities by Lowering the Barrier for Potential Undergraduate Researchers. So this is very different in scope. It's really focused on UVA students, undergraduates in engineering particularly, but I think that the goal to go more broadly past undergraduate it's just in engineering. And that, you know, the philosophy behind this, just briefly, is that experiential learning opportunities, particularly undergrad research, is really important for ensuring a seamless transition into the workplace or into graduate work or whatever the students go on to do after they graduate. And having these opportunities is I would argue far more important, honestly, or at least as important, but maybe more important even than the kinds of things that are learned in the classroom. And so the goal, though, is to try to get students into these kinds of research experiences earlier on. And I just would say that, you know, for those of you that attended earlier this morning session with Professor Barker about the large proportion of our students that have extracurricular experiences, I think one way that we've been successful is because they get early opportunities in engaging in research skills, and all of our advisors and our faculty are really active in reaching out to advisees and trying to encourage that. At least I know I am, even as early as during the first year. So here's an example of one way we do that, and I'll let Connor take over and talk about how we could try to systematize this a little bit more broadly.
Awesome. Thank you for the intro, Dr. Allen. Yeah, as Dr. Allen was saying, my name's Connor. This year I've spent my capstone creating Starting an Undergraduate Research Experience, or SURE. The program is designed to be able to facilitate student interaction with higher level research here at UVA. There are a whole bunch of reasons why it's difficult to become involved in research, not just at UVA, but at most public institutions. I know a lot of us have all been here, imposter syndrome, feelings of not belonging, and like the I don't know if I'm good enough to do it type thing. The program is specifically geared at trying to break that down, both by giving students marketable tools of like, oh, I can now do these lab skills and I know how to read a paper and write a cover letter, but also just to show that there's a larger undergraduate research community here that we can all connect to and be a part of. Also, of course, during COVID, it has been extremely difficult for anyone to get into labs, even like the grad students. So we wanted to try and provide some training modules to be able to help in the intermediate. Another is that students all come into UVA with different levels of experience. We have some people who took like all the AP courses that exist and other ones that haven't stepped foot inside of a chem and bio lab. We want to try and make it more equitable opportunities for all students to be able to enter interesting research opportunities during their time here at UVA. So what did we do? We created this program here to help interested undergraduates get research fundamentals. We recruited, this is all data from the fall, we recruited a wide variety of students from different years, a lot of first years, a lot of second years, so we try to make it as accessible as possible to younger students. We also wanted to make sure that the program was not biased to any major in particular. Of course, BMEs tend to try and find research opportunities wherever they can, but I was very, very happy to see that we have a large representation across all the majors. What we did is we had workshops with small groups in the middle, and this diagram shows the progression throughout the fall semester. We purposely had the fall groups led by current undergraduate students that have research experience to be able to make a smaller scale environment where people can get feedback and advice from their peers. You know, I got real with my participants a few times, like saying some of the difficulties I've had. And it's a real nice place for heart-to-heart moments if you like, which I think I did a little bit too much for my students, but they liked it, I think. We also offered a full credit course in the spring, trying to focus more on things in the laboratory. So this past semester, it was writing a grant from start to finish to show students how that process works. It's also all student-led. There's a lot of research out there showing that peer mentorship and peer learning are incredibly useful tools. So we wanted to leverage that as much as possible. And we also provided a lot of optional assignments, mock interviews, and other things for students to try and exhibit their newfound skills. And overall, we, along with trying to make students find research more accessible practically, we also wanted to feel more accessible emotionally by increasing belongingness, by providing the resources, providing a peer network, and overall making students feel like they can do it because we've all been there before. Yeah, I'm happy to take any questions during the question time. Thank you all.
Wonderful. Thank you so much. Thank you all the teams for presenting. Those are really exciting projects. And like Dr. Allen said, really timely projects. So now's the Q&A time. So for all the audience members who have a question, you can either unmute yourself and ask it, or you can put it into chat and I'll propose the question. Just think about what you want to learn from these teams. You know, how did they come about with their projects? What resources were available to them, et cetera, et cetera. So think about it for a second and then ask your question. I can actually get us started with one, and that way when they're answering that, you can be thinking more about what you would like to ask. So can each of you, the four of you, talk a little bit about how you went about choosing your project? So what does the project selection process look like, and why did you choose what you chose? Maybe we can start with Hunter.
Sure. So I did the capstone. We had like a quick interview round robin kind of session that our wonderful professors set up in replacement of like an in-person sort of thing. And so you just kind of sign up for the projects that sound interesting to you. They provided us with descriptions of each project beforehand. And so we like all got to read through them and sign up based on what we thought we would most like to participate in or what we'd be good at. And then we just kind of hopped to Zoom rooms and talked to the advisors of the projects and got a little bit more information about the projects, and they got to learn about what we do and our skills. It's kind of like I'm good at that, you know, you pitch yourself very quickly and as conversationally as you can. But I personally chose my project because I am a double major in anthropology, and so structural inequalities are something I really love to learn about, and particularly in Brazil, I've done a lot of previous work on that. And so especially around maternity in general, and I think something about that particular subset of the population is very intriguing to me and how they get overlooked. And so that was awesome, and it just kind of aligned with all of my interests, and I want to go into public health after this. And so it's a very great project to be on, to learn so much from these people who work with this group in Brazil and from our advisors. And so, yeah, that's pretty much how I found mine. My wonderful advisors picked me and Kunal and Simone, and yeah, it's just been great. So
thanks, Hunter. Maria?
Sure. Mine was kind of an untraditional route, since everything was going to be online anyway, at least for the beginning half of the year, most of it anyway. I figured I would just reach out to this lab in Texas because I really wanted to do women's health research. And in particular, this lab does like vaginal wound healing and has actually done some engineering stuff with creating like vaginal stents. So that was really exciting to me. So I just reached out to them, and my advisor responded and was like, I have no shortage of ideas for innovations in women's health, and kind of went through a list of like cool ideas. And this was one of the Vagina-on-Chip was one of them. And I thought it was a really cool way to combine like my passion for like women's health innovation and applying all the things that I've learned in all my BME classes. So it was like the perfect combination, and it worked out pretty well. I will say that I'm also getting help from a lab at UVA that does work with microfluidics. And one of the grad students in that lab has helped me to 3D print my prototypes. So I've been using some of the UVA resources, as well as getting like mentorship from the lab in Texas.
That's great. I think it's a good example of how our students think outside the box, right? And I think the culture allows for that and embraces that. And so if we explain that things should happen this way, students are free to say, well, what about this? And then they can pursue that, which is great. Erin?
Yeah, so I kind of sought out my advisor before I sought out a project, but I had worked with Dr. Tianucci before. She kind of connected me to a previous research project that I did in Tanzania, which was tracking the species of enteric pathogens there. And so I was like, I knew I wanted to do something with her similar to that project. So I just kind of reached out and was like, what are you working on? And can we be helpful at all? And so she's like, we're actually doing all this COVID stuff. And we're like, oh, wow. And then we kind of had to brainstorm how we could be most helpful to those projects when we couldn't be in the lab and doing the like molecular work that she does. And so we kind of used our BME coding and all that experience to provide kind of a out of lab assistance to that project. So yeah.
Thanks, Erin. Connor?
Yeah, so I actually have been sitting on Sure for a while now. Among all my younger BME friends, I was that guy that they came to for advice about research opportunities. So I saw that there was a need here, and I'd actually been working on this with Professor Helmke, my advisor, for a little bit before it. And I was really fortunate that he was okay with turning this into a capstone project too. So it was really cool to petition the BME department to say that there's this cool thing that I wanted to do. Would you support me through this for the next year?
That's great. Are there any questions from the audience? Okay, keep thinking about your questions. I'm gonna pose another one for the group. So maybe one is how it's a two-part question. How do your BME curriculum prepare you for your capstone project? And then what resources available here in BME and at UVA or in engineering or outside were available to you as you worked on your project? And we'll go backwards, start with Connor.
There are a lot of resources. My advisor is the director of undergraduate research into engineering, I believe. So there was a lot of access to surveys that had previously been conducted among UVA engineering students, a bunch of other people in administrative roles that I was able to talk to, like at the Career Center, the Center for Diversity Engineering and such. It's an overall really awesome community that if you have an idea, everyone will run with it, which is great. Classes particularly, I don't know, I think they just give you a good mindset for finding interesting problems and thinking through how to solve them.
Thank you. Erin?
Yeah, sorry, I would say mostly the computer science and coding backgrounds mostly helped for this project, which is not something I expected to be working on, but definitely helped prepare us because that's pretty much, yeah, all we've been doing to develop the dashboard. And I think also just the cell biology and basic biological background helps as well. But yeah, definitely the coding.
Maria?
I would say a lot of different things. The work in CAD, so like designing the 3D prototype, we learned those techniques second year in our design and discovery intro BME kind of course. So we had some experience with 3D printing already, which was helpful. Then just basic like the biology background of like cell interactions. It was really helpful because in the fall I was taking for my BME elective biomaterials, and then now I'm taking tissue engineering. So it kind of perfectly aligns and has been helping me throughout this project as I learn about like different techniques and ways of using hydrogels and just like recapitulating those like 3D cell structures.
Thanks. Hunter?
Similar to, I think, what Erin said. So our project is mostly, it's just computational. And so a lot of the coding we had to do in previous years definitely helped. Like we do a lot of MATLAB third year, and like we've in our project been using R and some Python, and we're exploring what using MATLAB would look like for the rest of our project. So it's been really nice just having like a basic understanding of what's going on in that particular realm, because we get to use our biomedical knowledge in like a different facet, which is really cool. But yeah, mostly for us, just the computational background has been really nice, and knowing you know, cell bio, because we have to talk a lot about what pregnancy does to the body and what it looks like, and childbirth and things like that. So yeah, mostly that.
Thanks. All right, get some questions in the chat. One is, how has working on your capstone fit into your schedule and classes? Any volunteers to tackle that one?
Sure. Capstone is a class, so that's been really nice because we have like one-on-one check-ins. We do smaller assignments to keep us on track. And then it's mostly, I think after third year, you just learn to work better in groups and schedule group meetings a lot better after Ideas Lab. And so it's really just everyone pitching in to meet and discuss. And you're supposed to do, I think, around like 10 hours a week is what it is. And like sometimes, you know, like that just doesn't happen. Sometimes it's way more. And so it's just, you hold each other accountable and you fit it in. And it's really not something that is like bothersome to fit in, because fourth year, like this is what you're doing your capstone and you're doing your STS thesis, your research paper and stuff. So everything kind of goes hand in hand, and it's always at the forefront of what you're doing as a student.
Thanks, Hunter. Anything to add for the other three students? Okay. All right, another question is, what has been the most tedious part of your project and how did you overcome that obstacle? That's a great question. Who wants to volunteer to answer that one?
I can talk a little bit about some of the problems I faced while prototyping. So originally I had two different prototype designs. So one where the one I showed you where the hydrogels are vertically stacked, but also a design where there would be channels where the layers could be like next to each other. I actually have like the little prints here to show you guys. If it's kind of hard to see, but like to see how tiny they are. But because they're so tiny and because like these little channels are so hard to like load stuff into, once we printed them, the first thing we had to do to test if this could even work is like we used agarose, a gel, to load into the wells and see like if there's leakage, if it actually can stack together. And so that was kind of tedious because the horizontal design didn't really work, and we would try to adjust the dimensions, but like agarose was just spilling everywhere, and like the guides we put in didn't really work. So that was a little bit tedious, but like, again, part of the design process. And that's kind of why we pursued two different designs. So yeah, that's it.
I think that's why these kinds of real world hands-on projects are so important, because that is the design process. That is engineering, right? It's trial and error, it's failure, and then overcoming that failure. Any of the other students want to comment on this question? Most tedious part of your project?
Our most tedious part of our project was just having to learn a whole new coding language, and then being able to, yeah, I guess learn all the different packages and how to best use them and represent our data well and accurately. But yeah, we spent a good like month, or not month, but at least like 2 or 3 weeks learning how to do everything, and then we could really be able to implement everything. Yeah, we do all ours in R, so.
Connor or Hunter?
Yeah, I'll just throw in there, our most tedious part, we're working with a very vulnerable population. Like we're working with data on pregnant women, and that in itself is like very hard to get stuff approved for, which is why there's like a lack of data and representation in general. And so the most tedious part of our project has been doing the IRB grant. We submitted it last semester, and like I'm still doing iterations for it because the reviewers just send back comments, and it takes like 3 weeks, and then they give you more comments and you have to turn it back in, and then you wait 3 weeks. And so like, it's been a really long process to try and get data. And so we've had to like exclude part of our project because we don't have enough time. We were originally going to do like a Virginia spread model, and like, we just don't have access to that type of data because we can't get it in time. And so we've been using like public health available data in Brazil through like the data SUS, their universal healthcare system and things like that. So we've just had to find other avenues of getting the information that we want, because it's just been taking a very, very long time. So
thanks, Hunter. For those that don't know, IRB, it's Institutional Review Board. So that's something at all major research universities, and it's there to protect human subjects research. So any humans involved in research at all. And it makes sure that they are protected, their privacy is protected, that they're not being coerced or mistreated in any way. So they're really strong protections that need to be there, but because they're there, it can be a little difficult, right? But it's supposed to be difficult because it's human subjects in research, but for sure it's tedious. Connor?
Less tedious and more time consuming. Back in the fall, there were 120 students and I had to pair them into 15 small groups based off their research interests. So I had to read through literally like a mini essay from everyone. And I know the two professors here going like, oh, it's no big deal. We read through all those essays all the time. But for me doing it for the first time, it was a long process. But thankfully it got done.
All right, great. Keep putting your questions in the chat. I'm going to ask another question to the group of students. How do you think your capstone experience will help you in your career? What are some tangible ways that this experience will help you get your next job or the job after that? Maybe we can start with Erin.
Sure. I'm actually planning on working for my advisor next year. So I'm really excited about that. And I think infectious disease surveillance is really something that I want to do for my career, especially in developing countries or low income populations, to be able to identify the different species of pathogens that are circulating and then be able to target interventions that are specific to those pathogens. So yeah, I think, yeah, this set me up nicely for next year because I'll just be continuing to help her with her projects. And she's also expanding to Pakistan and to India, into the refugee camp in Bangladesh. So it'll be good to be able to continue helping in those areas as well.
That's wonderful. Hunter.
So I kind of mentioned earlier, like I want to go into public health. So something that I really want to focus on is just trying to reduce the inequities in between groups of people that are created by structures of oppression and systems of oppression that cause like health crises, and trying to do so in a way that is not harmful. And so something about our project right now is like doing the research on how to make things actually beneficial to the community and not just impose whatever we think is best. And so I think that's been like really valuable. And I think like a lot of even the coding experience will be very helpful, like learning how to model disease spread and things like that is very useful for these types of health interventions and stuff like that. So yeah, it's a good background. I'll say that.
Thanks. Maria?
Yeah, so I also might be working for my advisor after I graduate. I think the most applicable thing, and I think it's something that we've had to do as part of the class, is write a grant. And that was kind of like, or a grant format assignment. And that really introduced me for the first time for like how to write a grant. And after working more with my advisor, and now we have to write other grants, like it's really helped me like learn a new skill that I'm learning is so important for research. And I want to go into women's health research later down the line. So I think that's a really important skill that it's helped me to build.
That's great. Connor?
Yeah, I've always wanted to be a teacher, so this solidified in my mind my passion for going into like academia and stuff like that. I've really enjoyed the student interaction and the feeling of doing something that contributes to people's professional development and becoming a part of a community. So I hope to continue doing stuff like this in the future because it's been really fun and impactful.
I agree. All right, there's another question in the chat. So what do you hope to be the aftermath of your projects? So maybe the downstream impact of your projects. Who wants to volunteer to go first?
I can go first. Mine's probably the easiest because we just hope to do the same thing but better next year. Found some awesome younger students who are gonna take on the mantle and lead this next year. And very, very, very long term, it could be an accredited program, but for now we're just going to try and provide useful information.
Great. Anyone else?
Mine is, the end goal for mine is to create like a commercialized product that can be like widely used as a vaginal model for studying tissue. So we're like actually working on like a patent application right now, and like so that we can continue it further.
Erin or Hunter?
We are hoping to improve a lot of the poor birth outcomes for underrepresented communities in the pregnant population in Brazil, just by recommending better vaccination timing. And hopefully this model can be supplanted with other data from different places and used in a similar way for this population, which would be really awesome. And maybe somebody next year could do the Virginia model that we so wanted to do, and things like that. So
Great. Erin?
Yeah, and ours is obviously a little bit timely, but just trying to create better surveillance methods in developing countries, and hopefully if we can get the process out to other people, they might be able to use it for other developing countries and communities as well, to be able to track the prevalence of COVID. So.
Thanks. We're running short on time. Are there any last questions?
I'll ask one.
Great.
I haven't asked one yet. It's been a quiet group, given that we've got some, obviously some prospective students and possibly some others who are going to be watching recording of this later on, who are trying to decide maybe between UVA and other schools, or trying to decide what they should be doing to do the awesome kinds of things you're doing in your capstones. So put yourself back maybe 4 years ago. If you were listening to a similar panel, is there something you wish someone would have told you at that point that, obviously you've all been successful, but that you wish you had known coming in, or when choosing what school to go to, what major to go into, to set yourself up for a 4th year capstone? I know it's hard to think maybe retrospectively, but just something along those lines.
I would say, so I mean, coming into college, I really didn't know what I wanted to do. I had some research experience in high school, but I didn't really know if that's the path I wanted to take. But my piece of advice would be like, don't be afraid to put yourself out there. Don't be afraid to ask questions and reach out to people and just talk to people. That's like kind of how I discovered what I kind of want to do, is just like putting myself out there, learning about what's out there, being curious, and not being afraid to reach out and I see how sending that one email, not being afraid to just say, hey, do you have any interest in mentoring a student, even having no contact with them before, has turned into such a fruitful experience for me.
I would kind of just echo some of that as well. I would, I guess, really encourage you to seek out what kind of health problems or biological applications speak to you and fuel your interest and your passion, and kind of capitalize on those. And UVA is just like, in the BME department in itself, is just a wealth of resources for that. So yeah, I would say seek out what you're passionate about, and to be able to like use your background for that is a really cool opportunity. So yeah.
I kind of wish, so I completely, yeah, everything that Erin and Maria said, totally on point. And like UVA, your professors are definitely here to help you. And like, don't be afraid, even in big classes, like everyone just wants you to be successful and find what you love. And so that's one thing that UVA is really great about, is like community, especially in BME, like everybody just wants to help each other, which is really, really awesome. And also I will add our 3rd year lab class. Everyone talks about it like it's really scary and it's really hard and it takes a lot of your time. And while yes, it does, it's also like, it's my favorite class of all of BME. It was so helpful. It's really enjoyable. You learn a ton, especially if you haven't been in a lab before. And it gives you a lot of really good like group work experience that definitely helps you for your capstone. And so don't be afraid of everyone telling you that BME is hard, because you can do it. If you are interested in it, you can do it and you will enjoy it. I promise. It's a really great major.
I think what's helped me a lot is making time for myself, doing things that I enjoy. If you like playing an instrument, if you like doing art, if you like reading, gardening, whatever it may be, make sure you find time to do it even when it gets super busy. It's great to keep your sanity and mental health. It's incredibly important. So whatever you find enjoyable, always make time to keep doing it.
Well, that was excellent advice from all four of you. Thank you. I've put my contact information in the chat. If anyone else on the call wants to put their information in the chat, it's completely optional. But you feel free, if you have any questions that weren't tackled thus far in this conversation, please feel free to reach out to me or whoever else shares their contact information. Dr. Allen, you directed that to me, so it's okay. All right. So
I realized that as soon as I did it.
All right. Thank you all so much for coming. Thank you to the students for talking about their projects and for sharing your wisdom with that on the audience. We really appreciate it. Everyone else, have a great rest of your weekend. Bye.
Thank you all.
Great to
Thanks,
see
everyone.
everyone.